Alrighty, then I'll get started. So I'm Tessa Hochhaus. I'm a PhD student at Texas A&M in the Rose Genetics and Breeding Lab with Oscar and Dave. And today I'm going to be talking about a paper that I published this year. And Jeekin mentioned it earlier. It's a meta-analysis for rose rosette disease resistance, QTL, and then I did a candidate gene search. So just a brief introduction. I don't think I need to go over symptoms. We all talked about that enough. But a quick overview of the two papers that I included in this meta-analysis. So I have papers from Jeekin as well as Ellen Young, a past postdoc in our lab, doing both tetraploid on the one paper and then diploids on the other. So from the tetraploid side, they had QTL on linkage group 5, 6, and 7 from the tetraploids, as I mentioned, and then diploids 1, 3, and 5. So what I wanted to do with this meta-analysis, I can switch over my main objectives, were to basically integrate these studies to basically get a better understanding of the relationship between diploids and tetraploids. Are they co-localizing? Are they in the same location? And then by doing this meta-analysis, being able to narrow down those regions that also focuses our candidate gene search. So starting with the diploids. So Ellen had a multi-parental interrelated diploid population. It's a bit of a mouthful. There you see the sources of resistance. I wanted to make like a disclaimer. If I say resistance, it's more reduced susceptibility. And for phenotyping, it was from 2019 through 2021. We saw the severity scale on Dave's presentations. So severity, but they also phenotyped for virus presence as a CT value. And then here's basically an overview of what they found. So she used MapPoly and MapFuser for the linkage mapping, and then FlexQTL for the QTL scan. And if you see the QTL, basically if you see RRV at the beginning, that's for virus presence versus RRD is for severity. And then you can see the years versus overall. And then on the tetraploid side, for Jeekin's paper, we have two populations, the stormy weather by bright eyes and bright eyes by migro, which you can see off here to the right. And then phenotyping was very, very similar. There were a few differences. In 2021, we had two collections of data. So you'll see a 2021A and a B. So you can see that here. So again, some quick map statistics. For this one, using MapPoly for the linkage mapping, and then QTL Poly. He also did a joint analysis, so for the two populations together, and for that he used DiaQTL. All right, so we didn't want to just use these two studies. We also wanted to kind of give some input and also give some more like statistical power by including more QTL. So what I did was a remapping of these populations. The two individual studies used an MDS approach. I'm not going to get into like the details, but basically end result, we have all of these really small inversions down the chromosome. So we wanted to make better maps to input into this meta-analysis. And to do that, we used the hidden Markov model HMM chain with MapPoly. And that bases it off of physical order of markers. And then I use QTL Poly for the QTL scan. So there's just a quick overview. I got to add a decent amount of QTL to the meta-analysis. I did use the three largest populations for the diploids. The third one's not here. We didn't find any QTL for that population. All right, and then for the actual meta-analysis. So I use a software called BioMercator. Basically, it's a way of combining all of these maps together and then finding these meta QTL. So what it needs is a map file and a QTL file for each of the populations. So for the map file, you have general population information, as well as then just the marker names and their position, what linkage group, and so on. And then a QTL file where you give it the QTL name, the year, the location, and then where in the chromosome it's positioned. You can then create a consensus map within this software. You basically choose a reference, and then it continuously adds the markers for each of these populations until you have one large iterative map. And then you can go on to the actual meta-analysis step. So there's two methods that you can choose from. One is the Gerberg-Off-Net method, but we use the updated version of that, which is called the Verrieres method. And it has two rather simple steps. One is called cluster QTL. So it basically outputs the optimal number of meta QTL for each linkage group. And that's based off of lowest AIC. And then from there, you can go to the visual representation of that, where it actually shows you the meta QTL and their positions, which looks like this. So just to kind of clarify what you're looking at, off to the left, we have the QTL from the individual studies. And then on the linkage group itself, you'll see these colorful blocks. Those are the meta QTL discovered. And the color of the QTL correspond to the color of the meta QTL. There's one very obvious, interesting meta QTL, I think, and that's this one here. We called it meta RRD 5.2. It's the second meta QTL on 5, where we have this large colocalization of QTL from both the diploids and the tetraploids and the remapping, which gave us this really nice, concise QTL region that really narrowed down our candidate gene search for that one specifically as well. And then we also see not quite as intense, but still a large colocalization here on 6 for 6.1. And it was interesting because we also found from the individual studies that linkage group 5 had a large proportion of the phenotypic variation explained. So super interesting. This is just giving you the numbers. Again, we have this really nice concise region here for 5.2, both roughly for centimorgans and also roughly for megabase pair. Before we did the remapping, that was not the case. We had like a real large discrepancy between megabase pair and centimorgan. So it was really nice to get that fixed after the remapping and mapping it by physical order. All right, and then we moved on to our candidate gene search. I just want to give a disclaimer. This is really like preliminary results. We're not definitively saying these are the genes that potentially are causing this reduced susceptibility, but it's a great point to start. So things that I looked for was a manual search in the Rosa chinensis reference genome. And we looked for antiviral mechanisms. The next two on this table are basically R genes. This next one is an interesting one. I'll talk about it in a second, but it's viral mRNA translation or synthesis. So I'll get to that in a second. And then also just general defense response genes. So if we're looking at our two like highly interested meta QTL, we're seeing that 5.2, really nice small consensus region, and it also has that antiviral mechanism. So that's really exciting. All right, so what are these? Obviously, there's a lot of candidate genes, more than what we'd like, so I can't go into detail of every single one, but here's like a general overview. So for that antiviral mechanism, it's a homolog of ISG15. And specifically for 5.2, it's called NDC1-NUP, which is involved in the bidirectional movement across the nuclear envelope. There was a paper in Arabidopsis that had a very similar gene. It was called NUP160. And when they had a mutation in there, there was a decrease in the basal resistance, disease resistance. The next one that I already mentioned, those are R genes, can have many hits. I can't really go into like every single one. And then viral RNA translation synthesis. So this is more on the side of viral susceptibility, not resistance. Most of these were related to ribosomal proteins. So we know that viruses hijack these ribosomal proteins. It's not really within the scope that we want to look at these, but again, I thought it was interesting that it was within these regions. So I included it in these results. And as for general plant defense, these are kind of the main categories that I found within each of the meta-QTL, all related to infection, wounding, the penetration stage, and then we also had some accelerated cell death through the SA pathway. All right, so that's basically everything that we've done so far. And now what I'm gonna talk about next is what's to come. So I mentioned that our candidate gene search was within Rosa chinensis, which is our reference genome. So we basically had these regions of our newly found meta-QTL. I could then go into the Rosa chinensis genome and figure out what candidate genes are within that region. However, now we have the Bayes-Thornless genome assembly, which is more closely related to our populations. It's in the parentage of M44, which was on the diploid side. So what we want to do next is basically figure out where this region is within that assembly. Hence the question mark. We haven't done it yet, so we're interested in what that holds. And then within that region, again, doing the same process and figuring out what candidate genes are in that genome assembly. All right, and then once we have that completed, another step is that we want to apply these to other diseases. Before we get to this step, we'd also like to create better a consensus map. We weren't quite happy with how Biomarketer kind of combined them. So creating a better consensus map and then applying the same methodology for both black spot and Cercospora using the papers that you see here and the ones that Jeekin just talked about. All right, so just an overall summary. It's obviously really important to understand these genetic factors, considering that control is so difficult. I think the genetic way is probably our best bet, right? It was interesting to see how the tetraploids and the diploids co-localize, and we really got down to narrowing those regions significantly, especially the one on 5.2, as I mentioned, with that 4 centimorgan range that also contained our antiviral mechanism. We still want to do more work to further narrow those regions and then hopefully create a marker-based selection tool so that we can track those QTL in our populations. And then, as I mentioned right before this slide, see what candidate genes are in the genome that's more closely related to ours, and then apply that to our other diseases. All right, so with that, I'd say thank you. Thank you to all my co-authors and also for our sources of funding, and I'd love to take any questions.